Last lecture we had seen the types of immune system, isn't it? Now we are going to see the cells which are involved in the immune system. In the very first lecture, if you remember, I told you that the immune cells and organs were coordinatory. So what type of cells, where they are produced, what is their function, what is their shape, what type of system is involved in the production of immune cells, that we are going to see today. In our human body, the long bones are present, for example, femur. Okay. So this bone, long bone, consists of a central red region that is termed as the bone marrow. Okay. The bone marrow is a site present in the long bone, is responsible to produce the immune cell. How? Because it contains the hematopoietic stem cells. These are stem cells. But as the age increases, the bone marrow red portion is replaced by the fat bodies, replaced by the adipose tissue. So the red part which is present in the long bone consists of, says bone marrow, which consists of the homeopathic stem cells. These cells have self-regenerating ability. And secondly, they produce two lineage. Once the stem cell produces the lineage, it lose its self-regenerating ability. So what are that lineage? That are the myeloid lineage and the lymphoid lineage. Okay, so every stem cells is self-regenerating, and if it enters in the production of lineage, it loses its capability of self-regenerating because it produces a lineage. So when there is no infection, all the system works normally, the homeostasis is maintained. Means what? Enough number of cells are produced, red blood cells are produced, WBCs are produced, platelets are produced, and the system is going on smoothly. But as soon as there is the antigen attack on the body, the hematopoietic stem cells are provoked, proliferate, and they produce number of various cells to kill that antigen. The first lineage which is produced is termed as the myeloid progenitor, and second is termed as the lymphoid progenitor. So in bone marrow, stem cells produce two lineage, myeloid progenitor and lymphoid progenitor. Okay. In the myeloid progenitor, for example, this itself is get differentiated into the dendritic cells. Most of the dendritic cells are present beneath our skin. They have this pseudopods-like structure which are involved to capture the antigen if it enters from the skin root. Once it enters in the skin, from the skin, these cells, dendritic cells, phagocyte it and process it, produce phagosome, process it, and this dendritic cell then with that antigen goes towards the cell, T cell. Okay, so this dendritic cell at that time when it carry antigen, it is also called as the antigen presenting cells. What are lysosomes? Lysosomes are the suicidal bag of cells, or you can say that this is a bag which contains many of the proteolytic enzymes, nucleolytic enzymes. Many enzymes are present in the lysosome which act at the acidic pH. So with the help of these lysogenic enzymes, the antigen which is trapped here, it had get digested. A part of that particular antigen has been presented by dendritic cell to the further to the tissue cell. Now this myeloid progenitor is further differentiated into monocyte and you can say that the granulocyte monocyte progenitor. Okay, so granulocyte monocyte progenitor gives you the monocyte and neutrophil. Why it is termed as granulocyte? Granulocyte means what? It contains granules. Therefore, it is termed as the granulocyte. So, neutrophil progenitor gives neutrophil. Granulocyte progenitor gives granulocytes. Okay. See, this is also the granulocyte. Okay. But in granulocyte, there are different cells. That is which contain granules. For example, monocyte contain granules, basophil contain granules, eosinophil contain granules, neutrophil contain granules. So neutrophil progenitor gives neutrophil, basophil progenitor gives basophil, eosinophil progenitor produce eosinophil. So such type of progenitors are differentiated from the myeloid progenitor. Okay, do you remember this? So main is myeloid progenitor, which gives you the progenitor of the granulocyte cells, and from the granulocyte progenitor we will get the granulocyte. So this monocyte is contained, that is a big cell. What is a nucleus? Is like the kidney shaped nucleus. It also contains a lysosome. It has a capacity, the monocyte has a capacity to enter into the cell. 5 to 10% of WBCs are from the monocyte, to enter into the tissue after infection, and itself is get differentiated into the macrophage. Okay, so monocyte itself get differentiated into the macrophage at the infected sites, and this macrophage is 5 to 10 times larger than the monocyte. The nucleus is more, it contains the lysosome. The lysosomes are responsible to this, you can say that the digestion. This macrophage has the pseudopoda-like structures which are responsible to capture the antigen present at the site of infection or at the site of inflammation. After capturing, the antigen undergo process by the lysosome, phagocytosis occur, lysosome, then it produces the phagolysosome. So besides this, macrophage are responsible to secrete the inflammatory cytokines IL-1, IL-6 and the tumor necrosis factors. So it also responsible to produce the reactive oxygen species and reactive nitrogen species. Reactive oxygen species means what? It has some extra energy. Okay, so that reactive nitrogen and oxygen species are responsible to kill the DNA, protein which are present in the, or many other biomolecules which are present in the antigen or bacteria. And in this way, using this, the macrophages are responsible to kill the antigen. The detailed structure we are going to see the afterwards. So remember, all this is the cell. So what is the function of this macrophage? Macrophage has lots of function. First is clear the antigen, clear the bacteria. Macrophage also present part of the antigen to the T helper cell. So that T helper cell then can initiate or activate the beta cell. Beta cell then produce the antibodies in response to the antigen which is presented by the macrophage. It also regulates the function of the cell properly to clearing most of the antigen foreign bodies which are present in the cell. Myeloid progenitor, in that neutrophil progenitor produce neutrophil. Neutrophil has the 50 to 70% has the neutrophil. Neutrophil contains trilobe structure. This is the nucleus of neutrophil. 50 to 70% of the circulating WBCs are nothing but the neutrophil. Neutrophil present is the first defense response by the WBC. Immediately the phagocytosis occur. It is a phagocytic cell. It can initiate the phagocytosis process at the site of infection. So this neutrophil, once is synthesized, it is circulating for 7 to 10 days. At the site of infection, it enters into the tissue, resides there for a few hours, and the secretion occur. This also, how neutrophils goes at that site? It because of the chemokines which are secreted at the site of inflammation, and because of this chemokines, neutrophils goes at the site of infection and start the phagocytosis and release some proteins so that inflammatory response will start it. So in this way, these 4 cells are responsible to activate the immune system, or this is the 4 cells which are there. Further cells we are discussing for that immune response.